thank Mafalda and Sonia for inviting me to participate in this virtual Submit SOLTI meeting. Just a brief comment, interesting comment. I was in 1994 at Memorial Sloan Kettering with Jose Baselga, who was running the first phase 2 trial with trastuzumab. And I had firsthand notice from him of how Herceptin was working in the first patient. And I just want to remember that the rate of response to single agent trastuzumab in the early 90s was only 10%. And the drug was about to be killed, but he himself and others did a tremendous job, and now the HER2 field is alive thanks to the view of him and other people at that time at Memorial and Sloan Kettering mainly, and other sites too. Today I will review with you the new horizons in the treatment of HER2 positive metastatic breast cancer. There are three aspects that I want to share with you. First is that today less patients have HER2 positive metastatic breast cancer thanks to the recent therapeutic advances in the early disease in the neoadjuvant and adjuvant setting. However, the flip side of these advances is that the patients that have recurrent HER2 positive metastatic breast cancer might be harder to treat than in the past due to a selection of patients that are refractory to neoadjuvant anti-HER2 drugs, and also due to the higher incidence of brain metastasis as a first site of relapse. Today, I will review how novel anti-HER2 drugs can help to overcome this situation today. Some of them already approved by the FDA, one also in China, and also review the promising results and plans with novel drugs and combinations. We have to keep in mind that despite all the advances that we have seen in this tumor today, HER2 positive metastatic breast cancer remains incurable for the vast majority of patients. So there are a number of unmet needs. One is to prevent and treat better brain metastasis. Second is to make metastatic breast cancer a really chronic disease, a disease that patients die with this disease, but not die for this disease. This, I think, should be a goal. Now, we are very happy with the 5-6 years of median overall survival, but for the patients is clearly insufficient. And of course, the goal should be to be able to cure the metastatic disease in the future. This year, we are living very important rapid changes, particularly in the United States, where three drugs have been approved in the last 12 months, particularly in the advanced setting in HER2 positive disease. In particular, tucatinib, trastuzumab, deruxtecan, and neratinib have been approved in patients treated with anti-HER2 therapies. I will review these four big blocks of novel therapies in HER2 positive metastatic breast cancer. And first, I will discuss the antibody margetuximab. The development of this antibody has been led by Hopriugo, so it's a pity that she is not longer here, but she's been the real leader of this drug. As you know, margetuximab is a monoclonal antibody that has been modified from trastuzumab in a manner that is more able to engage natural killer cells to induce antibody-dependent cellular cytotoxicity. And this has been achieved by modifying the constant fraction of the antibody in a manner that has more affinity for the CD16A receptor. The receptor is expressed in the natural killer cells and is the one that mediates ADCC. In addition, margetuximab has also the potential to improve adaptive immunity. It's important to know that the CD16A receptor expressed in natural killer cells has different genotypes in humans. And one of them is the one that has more affinity and more ability to induce ADCC. It's the valin-valin genotype. but the majority of humans have other genotypes that result in low binding affinity for antibodies such as trastuzumab. So margetuximab has the potential to improve the efficacy, particularly in these genotypes with low affinity. The phase 3 trial with this drug that was treated by Hobrugo, as I mentioned, was performed in HER2-positive advanced breast cancer in patients with at least 2 prior lines and for HER2 disease with an average of 1 to 3 prior treatment lines. The phase 3 trial compared in patients in this pretreated population chemotherapy with margetuximab or chemotherapy and trastuzumab. And the median PFS was in favor of margetuximab with a hazard ratio of 0.59, but the absolute improvement in median PFS was very, very modest. When the genotypes that are less able to engage ADCC that I mentioned before, when this subset of patients were analyzed in exploratory PFS analysis, the benefit was slightly better than in the overall population with a hazard ratio of 0.52. Until now, we have 2 interim overall survival analyses. In the first one, the hazard ratio was 0.95. In the second interim analysis reported at San Antonio, the hazard ratio was 0.782. And we are now pending on a final overall survival analysis to see if there is whether it emerges a population that can truly benefit from this antibody. Of note, as it was shown in terms of PFS in this second interim analysis, when the population of patients with CD16A genotype with low affinity, the benefit appeared to be numerically higher than in the overall population, but still not significant. Nonetheless, this antibody is under review by the FDA for approval. Now I move to 3 drugs that are potent tyrosine kinase inhibitors. The first one, neratinib, a drug that has the phase 3 NALA study that you all know has been led by Christina Saura. It's a phase 3 trial in patients with advanced HER2-positive disease who had 2 or more lines of prior HER2 therapy for metastatic breast cancer. And patients were randomized to lapatinib and capecitabine or neratinib, capecitabine, and prophylactic loperamide. The results of this trial were positive with improvement in PFS, with a significant improvement in PFS, with a hazard ratio of 0.76 and a significant p-value. Of note, the percentage of patients that had prior trastuzumab, pertuzumab, and TDM1 was about 1/3 of the patients, and brain disease was present in 16%. This is important to keep in mind these numbers because then we can compare this with the HER2CLIMB trial with tucatinib. This drug, based on the result, has been approved by the FDA and will be filed for EMA approval too. Trial that was presented at ASCO this month is the phase 3 trial with pirotinib, a drug that has been developed in China. And it's a study performed also in metastatic HER2-positive breast cancer in patients previously treated with trastuzumab and taxanes and anthracyclines. However, it has to be noted that the population is quite different from other studies in terms that the prior trastuzumab, pertuzumab, and TDM1 was not, these therapies were not exposed to patients in this trial, and the patient did not have brain disease. Also, the incidence of grade 3 diarrhea was 31%. Despite of these limitations, the drug had a very significant improvement in PFS with a hazard ratio of 0.39. With a median PFS that was 6.8 in the lapatinib combination. There was the control arm in the pirotinib capecitabine combination was 12.5 months. Of note, the overall survival results are not yet mature, but are also really promising. And this drug has been already approved in China And they mention specifically that not all countries have access to pertuzumab TDN1, and there is also room for these agents in these kind of populations. Now the trial that it's a practice changing trial once tucatinib is approved, as is already happening in US. The HER2 CLIMB trial presented at San Antonio and published in the New England Journal of Medicine in HER2-positive metastatic breast cancer. This trial has a very unique and robust design in the sense that all patients had prior treatment with trastuzumab, pertuzumab, and TDN1, and also the patients had a brain MRI at baseline. This is important because the trial allowed to enter patients with untreated brain metastases not needing immediate local therapy, and also patients previously treated for brain metastases who were not needing immediate local therapy. Patients were randomized to trastuzumab, capecitabine, and placebo, or capecitabine, trastuzumab, and tucatinib. The study was the population of the study had a high incidence of brain metastases, 48% in the tucatinib arm and 46% in the placebo arm, which I think is quite important, as was also mentioned in the prior presentation, that these patients need to be included in the trials because they are a reality and they need to have access to the best treatments available. The study was positive in terms of PFS and overall survival. The median PFS had a hazard ratio of 0.54, the median overall survival 0.66. The median PFS and median overall survival were not as good as in other trials, but it's important to emphasize that this was a heavily pretreated population with a high incidence also of brain metastases. The tolerability of the combination appeared reasonable, as only 6% of the patients discontinued therapy in the tucatinib arm due to adverse events. It was important in this study to know what was happening in the half of the patients with brain metastases, and we can see that the median PFS was also highly in favor of the tucatinib arm with a hazard ratio of 0.48. It's important to note also that in the control arm, there were no patients with brain metastases free of progression at 1 year compared to 25% in the tucatinib arm. As a result of all of this, this drug has been approved by the FDA and is under review by the EMA. Of course, we want to see whether this drug can have activity in earlier lines, and the more robust trial that is ongoing is the HER2 CLIMB-02, comparing TDN1 head-to-head with TDN1 plus tucatinib. Now I move to the third portion of the new HER2 drugs, and consisting of the new antibody drug conjugates. There is a particular interest in, of course, in trastuzumab deruxtecan, that based on the phase 2/1 trial presented at San Antonio and published in New England, has been already approved by the FDA. Trastuzumab deruxtecan was tested in a phase 1/2 trial in a very heavily pretreated population of patients. The median lines of therapy in this population was 6. All patients had prior exposure to trastuzumab and TDN1, and 66% to pertuzumab. The anti-tumor activity is really impressive with this single agent therapy in this heavily pretreated population with a disease control rate of 97% and an overall response rate of 60.9%. Also, 11 patients out of the 168 patients achieved a complete response. But not only the response rate was so high, so impressive, but also the median duration of response was very long, with a median duration of response of 14.8 months, a median progression-free survival of 16.4 months, and the overall survival not yet reached. There were a few patients in this study with brain metastases, and in the median PFS in this small population was 18.1 months, so suggesting also activity in this clinical setting. The drug appears well tolerated in general, but it has to be noted that some patients develop interstitial lung disease that can be important and with grade 3 and grade 4 and 5 cases. And it's important to be aware of this potential toxicity and be very strict in the measures of detection and management. Regardless of that, and taking into account the impressive activity in this phase 1/2 trial, particularly in the phase 2 part, the FDA has already approved this drug and has been filed also for approval at EMA. Again, what we want is to have these very active drugs in earlier settings, and there is the ongoing phase 3 Destiny Breast 03 trial comparing head-to-head trastuzumab deruxtecan with TDM1, and also there is an ongoing trial in a phase 3 trial comparing trastuzumab deruxtecan with investigator choice in patients pretreated with TDM1. There are many antibody-drug conjugates, but one that is also in advanced stage of study is CYT985. The phase 1 trial was led by Cristina Saura, as well as the TULIP phase 3 pivotal trial that is comparing in pretreated HER2-positive advanced breast cancer CYT985 to physician choice. So we'll see whether this also adds to the new drugs for breast cancer therapy. And now we enter in the last part of the presentation, reviewing some promising combinations of HER2 drugs with other mechanisms of action. Of course, one of the obvious and interesting combinations is in the estrogen receptor positive subset of HER2 positive breast cancer, the combinations of CDK4/6 inhibitors with anti-HER2 drugs. We already have some results, and one of the studies that has been already published is the Monarch-HER trial in patients with HER2 positive, hormone receptor positive advanced breast cancer, patients that had at least 2 or more lines of prior directed anti-HER2 therapy. There were 3 arms. The control arm will be trastuzumab and chemotherapy. The 2 experimental arms, abemaciclib and trastuzumab, or abemaciclib, trastuzumab, and fulvestrant. This arm, the triple therapy arm, had longer median progression-free survival compared to the standard chemotherapy and trastuzumab arm, and also a very interesting response rate of 32.9%. So this opens the door to have chemotherapy-free options for hormone receptor positive, HER2 positive advanced breast cancer patients progressing to prior therapies. Now I want to discuss a very interesting approach for further exploiting CDK4/6 inhibitors in HER2-positive disease in a very biomarker-driven way. And there are 2 lines of evidence that point to the interest of this approach. One is a very recent paper published in Nature Communication by the group of Aleix Prat, where they showed in cell lines and also in clinical samples that HER2-positive breast cancers, when they shift to luminal A intrinsic subtypes, they become very sensitive to anti-CDK4/6 inhibitors. So there is preclinical evidence of the interest of these drugs in these luminal A intrinsic subtypes. But also there was a trial, Patricia, presented by Eva Ciruelos at San Antonio 2 years ago, in which ER-positive, HER2-positive patients were treated with palbociclib, trastuzumab, with or without letrozole. And in those patients with luminal intrinsic subtype, defined by PAM50, the disease control rate at 6 months was 72% with palbociclib, trastuzumab, with or without letrozole. And this compares with only 25% in non-luminals. So based on this evidence, Solti has designed a very interesting study in hormone receptor positive, HER2 positive, but specifically with PAM50 luminal intrinsic subtype. These are heavily pretreated HER2 positive breast cancer patients that are randomized to palbociclib, trastuzumab, and endocrine therapy. versus physician treatment choice. That's offering here also the possibility to act for a chemotherapy-free option in advanced HER2-positive, ER-positive breast cancer. Now I will review just a few designs that advance biological therapies to frontline to first-line therapy, and they use the approach that patients are now treated in first-line, the vast majority with taxane or vinorelbine with anti-HER2 therapy, trastuzumab and pertuzumab. And after 4 to 6 cycles, the patients that have stable disease or response are randomized to maintenance of trastuzumab and pertuzumab or adding biologically driven option. In this study, the PATINA trial, patients in this maintenance phase, they are treated with anti-HER2 therapy and endocrine therapy or anti-HER2 therapy, endocrine therapy plus palbociclib. So it will be interesting to see if we can prolong the efficacy of anti-HER2 dual blockade in first line. Another ongoing design, I think, is led by Sonia Pernas. Maybe she can explain better where we are here. But it's a study in 2 parts, and this is the design of the second part, the EPIC-B2 trial, in which patients, the same approach as the study before, patients receive induction chemotherapy and dual HER2 blockade, and patients that are stable or responding are randomized in the presence of PIK3K mutations to trastuzumab, pertuzumab, and placebo, or trastuzumab, pertuzumab, and alpelisib. The SOLTI group is also doing a similar study with the AKT inhibitor ipatasertib with pertuzumab and trastuzumab in the maintenance phase, and with a very strong translational component that is also very important to see how this goes and all the data that derives from this kind of approach. Now, the last part is to review briefly the potential role of immunotherapy in HER2-positive disease. We have mainly the results of 2 trials, the Panacea trial that has been already published of pembrolizumab and trastuzumab in heavily pretreated HER2-positive disease. In terms of PFS, the results were poor in both PD-L1 positive and PD-L1 negative patients. In terms of overall survival in PD-L1 negative, the median overall survival was 7 months in the PD-L1 negative and not reached in the PD-L1 positive. However, it's important that in the PD-L1 positive patients, the response rate was 15% with some durable responses, so suggesting a role for PD-L1 in selected patients for further studies. In the PD-L1 negative population, there were no responses. The K2 trial is a randomized phase 2 that randomized patients to TDN1 and placebo, or TDN1 and atezolizumab, and also points in the same direction than the Panacea trial, that the benefit can be restricted if confirmed in further studies in the PD-L1 positive population. The overall survival data in PD-L1 negative patients doesn't show any hint of benefit, but in the population that is PD-L1 positive, there is a numerically superior progression-free survival on 1 year that is not statistically significant, but again points to a potential benefit in this subset. So this gives ideas on where to go in the research in this area. Of course, we want to see data in first line, and there are ongoing trials, and just mention here one of them with atezolizumab or placebo in first line patients treated with taxane, trastuzumab, pertuzumab with placebo or atezolizumab. The study is ongoing, and of course, it's very important to see the results. Of note, there is no selection of patients based on PD-L1 status. So, to conclude, we see that the field of HER2 positive metastatic breast cancer is changing very quickly. This year, we are seeing mainly a change in the treatment of options for patients after TDN1 and pertuzumab. But in the design that I just discussed, we have the potential to really change options in first line, second line, and third line in the years that are coming soon. To conclude, there are big advances on anti-HER2 drugs, but we are still far from success. I think the cure is the goal that we have to keep in mind. The challenge that is not easy is to make immunotherapy really effective as a first-line option. I think is where the effort should be focused. If not possible, so we have to keep on doing the disease more chronic. You have better anti-HER2 drugs, better combinations, and keep also persisting on doing studies in biologically defined subsets to prevent or overcome resistance. And keep in mind that today maybe 40–50% have brain metastases when they relapse. And we have to incorporate the best drugs first and include anti-HER2 drugs able to treat effectively these brain metastases. With this, I conclude, and I thank you very much for the invitation and for your attention.
Joan, thank you so much for your excellent overview of all the drugs that are being developed in HER2-positive breast cancer. It's really exciting times for this type of breast cancer. We have time for a couple of questions. So my first question would be, so we are moving many of these drugs to the early setting, and as of today, patients with early breast cancer may be treated with trastuzumab, pertuzumab, and T-DM1 in the early setting. So which is your first line regimen if these patients recur despite all these drugs that they have received in the early setting? Are you going for, imagine of course that you have all these drugs available, which is not the case in Spain, but when eventually we'll have all these drugs available, what would be your first choice?
Well, I think that I can answer from a general perspective. If we can have access to all drugs anytime, any line, or if we are driven by regulation and also for reimbursement policies. Of course, the most the drugs that I would like to use frontline in a patient relapsing early in 6 or 12 months after they finish T-DM1 or adjuvant anti-HER2 therapy with trastuzumab or trastuzumab pertuzumab. I think that the combination of tucatinib, trastuzumab or capecitabine, or trastuzumab deruxtecan will be, I think, the most active choices. However, if the patient has had only anti-HER2 therapy with trastuzumab or pertuzumab, the relapse is 6 months or 1 year, then I think that T-DM1 is still a very good option. Or if the patient relapses after T-DM1, I think it gets very, very complicated, depends on the time. And as I said, if the drugs that you mentioned are available, I think it's an easy choice. If not, have to prioritize based on what we all know a patient journey, see what are all the options that we have, and try to prioritize based on efficacy, toxicity, and also depending on the country and resources, based on reimbursement strategies.
Yeah. So another question I have is regarding brain metastasis, right? You have mentioned that, and you showed the results of tucatinib. And one question that comes to me is, what should we do with this problem in HER2-positive breast cancer? So should we start to screen patients with brain MRI to give them tucatinib before? Should we move tucatinib to the early setting to prevent the appearance of brain mets, or should we just, keep the lines we are doing right now, and when brain mets appear, then use tucatinib? So I think, to me at least, lots of questions regarding brain metastasis were reopened when we saw the tucatinib data, and I would love to know your thoughts on this.
I think that now, for instance, in the Catherine trial, the patient that relapsed with metastatic disease, half of them have already brain metastases as the first site of relapse. So I think that this mandate for reconsidering brain MRI for these patients, I think that maybe all of us have patients that thanks to screening brain MRI for a clinical trial, we have identified oligometastatic disease or single brain mets. And for instance, I have a patient that now is 7 years since she entered in a TDN1 trial, had a mandatory MRI prior to entry, had a brain metastasis, had surgery, had also disease in other sites. We took a very aggressive approach with surgery of the brain mets, with radiation to the oligometastasis, and she's free of disease now for 7 years. So I think that we should reconsider that. And of course, as you mentioned, the efficacy seen with Ducatinib also calls for their study in the neoadjuvant or adjuvant setting, not on to prevent also the brain mets, which should be the main goal, rather than treat to prevent. I couldn't agree more with you.
Well, Joan, thank you so much. It has been a great overview of all these drugs, and thank you so much for joining us in the SOTI UpToDate meeting.
Thank you. Bye-bye.